My name's Jacob Koszulski. This is my Make a Splash video about horseshoe crabs. There are 4 species of horseshoe crabs still around today. Only one species, Limulus polyphemus, is found in North America along the Atlantic and Gulf coasts from Maine to Mexico. The other 3 species are found in Southeast Asia. Horseshoe crabs use hemocyanin to carry oxygen through their blood. Because of the copper present in hemocyanin, their blood is blue. Horseshoe crabs' blue blood is so valuable that a quart of it can be sold for $15,000. This is because it contains a molecule that is crucial to the medical research community. Interest in the horseshoe crab has grown over the last half-century due to the distinctive nature of its blood. The animal's main commercial value is based on a substance found within its light blue blood. Possessing an open circulatory system with no adaptive immune response, the horseshoe crab has survived through the ages by an innate immunity based on granular amoebocytes, which comprise 99% of its hemocytes. When these granular amoebocytes come into contact with an endotoxin, a cascade of defense molecules is released, triggering coagulation and a neutralization of the pathogens. The resulting clot effectively immobilizes the threat and prevents an infection from progressing beyond the wound. The unique ability of amoebocytes to produce an instantaneous, invisible reaction to endotoxins has driven commercial demand from pharmaceutical and biomedical companies to confirm drug and medical device safety using the Limulus amoebocyte lysate, or the LAL test, which has become the method of choice for endotoxin detection. In order to make these LAL tests, the horseshoe crabs are captured and bled. The blood is then collected and centrifuged to concentrate the amoebocytes. Water is then added to the packed amoebocytes, causing them to lyse and release coagulation proteins. The Atlantic States Marine Fisheries Commission, ASMFC, reported that in 2015, 559,903 horseshoe crabs were transported to biomedical facilities for the production of LAL. The raw materials for the preferred LAL test require careful extraction of blood from horseshoe crabs. Established methods entail introduction of a hypodermic needle placed directly into the exposed pericardial membrane of the horseshoe crab to draw from 50 to 400 milliliters of blood, depending on the sex and maturity of the horseshoe crab. The harvest and collection procedure for bleeding horseshoe crabs may appear straightforward, but there are significant risks posed to the crabs at various stages of the process, ranging from transportation and crab storage to the blood drawing itself. Horseshoe crab mortality rates following such harvesting range from 10 to 30%. However, these figures do not account for any further trauma or detrimental behavioral changes once the animals are returned to the ocean, nor the derivative population impact from the disruption of horseshoe crab spawning. Atlantic States Marine Fisheries Commission reports on horseshoe crab harvest mortality dates back to 2004. From 2004 to 2012, the number of crabs delivered to biomedical bleeding facilities increased from 343,126 to 611,827, or by about 78%, while total mortality correspondingly increased by 75%. The pandemic has been a tragedy, but out of this tragedy, large advances in the biomedical field have been made. In only about a year, a usable vaccine was developed, and with the increase of biomedical research, there is an increase in the resource needed, like LAL. I'm proposing to start aquaculturing Limulus polyphemus to help alleviate the stress put on the wild populations. Even if only 100,000 are grown in a facility, that is 16.34% of the total caught from the wild. One of the main challenges with aquaculturing these animals is that normally they are fed frozen seafood that is bought from warehouses after they've passed their sell-by date. This is the most cost-effective source. However, when the fish are thawed in the tanks, some of the nutrients are dissolved, which leads to the crabs becoming malnutritioned. My proposal to combat this is to buy the feed fresh, or also to aquaculture some bait fish along with the crabs. This will keep everything contained within the facility and help lower costs in the long run. The profit for growing this species is large. Will just take some time to develop a facility, but there's always going to be a need for them in the biomedical field, and this field is only going to grow. That being said, the obstacles in the way are pretty minimal and can be easily solved. And here are my references. I hope you guys enjoyed this video. I know I did. I definitely learned a lot more about these crabs than I thought I was going to, and I hope that you guys can take something away from this.